Hello, and welcome to today's session on experimental control. As always, before we begin, please make sure that you have the PowerPoint lecture notes printed and in front of you so that you can take notes along the way. To put today's conversation into context, we will remind ourselves of the research cycle. As we've said before, in the science of psychology, we're interested in behaviors, thoughts, and feelings, and we might identify one of these real-world phenomena for an area of research. In order to create our research representation, what we might do is abstract from the real world by engaging in operationally defining our terms and by obtaining an appropriate sample to create this research representation. And from that, we will engage in different kinds of methodology to generate some research results. In prior videos, we've talked a lot about observational methods, and we've talked about surveys. Today, we're going to be focusing on experimental methods. Once we have our research results, we engage in all different kinds of data analytic techniques, and in a future video, shortly after you see this one, we'll be talking about a particular kind of frequently used data analytic technique called a t-test. And this will help us generate some research conclusions that will hopefully generalize back to the real world. To put this further into context, what we can say is that true experiments, are going to land at this portion of this particular conceptualization of research varieties that we've seen before, different approaches to research. Remember that we have three different axes here, and today we're going to be focusing mostly on this axis, true experiments, and this would be one extreme on a continuum that goes from true experiment to naturalistic observation. We call this the control axis. And we call it the control axis because in a true experiment, we have complete control over an independent variable, a variable whose effect we seek to know, and specifically, we manipulate the variable whose effect we seek to know in a true experiment. By contrast, at the other extreme of this control axis, we have naturalistic observation, where we might, for example, put up a video camera and simply watch behavior and see what kinds of things we might be able to glean from that purely observational technique without any manipulation of an independent variable. We should also remember that true experiments can be true experiments regardless of their setting. So that is to say, on this axis, which ranges from field to laboratory, we can have a well-controlled and highly manipulated variable that happens to occur out in a field. For example, we can have a case where we might be interested in how animals respond to conspecifics of different kinds of colors. So we might, for example, look at how the behavior of an animal, maybe even an insect, might vary if one of the members of its species is colored red versus colored blue. I have a colleague in biology who works on these kinds of questions, and this person will take decoy flies or decoy insects of other varieties, and they will be differently colored. And this person will bring the differently colored animals onto a particular insect or near a particular insect and see what kinds of responses that we might get as a function of what kind of color the decoy is. This is clearly occurring in a field setting, and we have a lot of control over the independent variable here, maybe the color of the decoy as being red or blue, but this is an experiment even though it's occurring in the field. It's an experiment because we're manipulating the independent variable. In my own research, I manipulate independent variables very directly, but I do so in a laboratory setting. I often use sensory experiments where I might present stimuli for just 20 or 30 milliseconds, and I'm manipulating the stimulus duration while I'm in a laboratory setting. So we shouldn't confuse this axis, the amount of control we have over the independent variable, with the setting in which the study might be taking place. Likewise, we might have another axis that shows a different kind of variability. Here we have the role axis, and here we have scientist as a participant, and scientist as an observer. And once again, we can be purely in we can serve as observers as people are completing our studies, and those studies might have genuinely controlled independent variables. This is my own scenario in my own research where I am truly an observer, but I don't interact very much with my participants. I'm not interacting with them directly. I have the computer make all the observations for me, but those observations are occurring in a laboratory setting, and those observations are occurring while the stimuli are being manipulated very precisely according to various independent variables that I might have. So here's the axis along which we are discussing today, or that we will be discussing today. And here we're going to be talking about true experiments. So let's see if we can begin by motivating you to do some of the reading and see if you can answer these potential pop quiz questions. I'll read through the three of them, and then we'll see what your answers might be in class. So the first of the three is this. Identify the three conditions needed to make a causal inference. Second, in your own words, explain how confounding variables are related to the issue of internal validity. And third, in your own words, explain what is meant by an independent group's design. We'll let you stop the video, respond to those questions, and then we'll take your answers in class. Okay, welcome back. I hope you had the opportunity to respond to those three questions. I'm going to elaborate just on the answer to the first of those three questions to get our conversation motivated today. We've talked a little bit about this question of the three conditions needed to make a causal inference, and to summarize those, the very first one is what some people find a little bit counterintuitive, and that is that correlation is necessary in order to establish a causal relationship. Now, what confuses people is that we drill home the point in Intro to Psych that correlation does not imply causation, and that's true. However, correlation is a necessary condition for causation. So when we establish that we have a correlation, we're part of the way there, but we need to do other things as well. Another way of thinking about this is that some correlations are causal, but many of them are not. So we need to have that correlational foundation first before we can move on to ask other questions. The second condition that we would need to draw a causal inference is to have an appropriate time-order relationship. Specifically, we need to establish that the cause precedes the effect. In some correlational research, that's not possible. The example that we've given before is the example in which we can say that there's a strong correlation between the number of guns in a city and the crime rate in that city. Those two are very strongly correlated, and they're positively correlated. As one goes up, the other also goes up. The challenge is in understanding the time-order relationship, if in fact there is one. It could be that the number of guns is driving the high crime rate, or conversely, it could be that the high crime rate is driving people to go out and purchase guns, perhaps for protection. So the direction of causation is ambiguous, and that makes it impossible to draw a causal inference. What we need to do instead, if we're trying to draw a causal inference, is establish very clearly the time-order relationship, such that the cause clearly is preceding the effect. The third of the three conditions necessary for a causal inference is perhaps the most challenging, and this is the elimination of plausible alternative explanations. And that's what brings us to this slide. To say that one variable causes another, we must eliminate plausible alternative explanations. And in most of today's conversation, we're going to be talking about different ways to do that. Basically, a way to achieve a plausible inference about causation by ruling out other alternative plausible explanations. Okay, we're going to be talking about two different techniques. One is holding conditions constant, and one is balancing. Generally speaking, we prefer to hold conditions constant whenever possible, and we'll say a little bit about that now. A factor that remains the same across experimental conditions cannot be the cause of any effects that are observed. And we'll see if we can give you an example or two. As a sensory psychologist, I'm very interested in, for example, how briefly a stimulus can be presented and still clearly seen by a participant. So I might be flashing words on a computer program, or a computer screen rather, and I might ask if people can read what those words are, and maybe a word is flashed for only 10 or 20 or 30 milliseconds. Now I can run an experiment where I can see how accurately people respond to these very briefly flashed words, and I might have one condition where the stimuli are shown for 10 milliseconds, another where they're shown for 20 milliseconds, another where they're shown for 30 milliseconds, as an example. If it were the case that I ran the 10 millisecond condition early in the morning, and then I ran the 20 millisecond condition with another group of participants in the afternoon, and then in the evening I finished the experiment by running my third group of participants with the 30 millisecond condition, it would certainly be true that I've distributed my conditions across different participants, but what I haven't done was hold time of day constant. In this particular case, I ran one condition in the morning, one in the afternoon, one in the evening. Okay? So it could be the case that maybe I'm going to observe some different performance levels in these three, but not because of something that I've done with my independent variable, the variable of interest, 10, 20, or 30 milliseconds, but rather because I have this confound, I have this systematic covariation between my variable of interest, which is the stimulus duration, and the time of day. So I'd want to be able to hold that constant, time of day constant, so I can draw a more plausible inference about the variable of interest. In this particular case, that would be the stimulus duration. Okay? So whenever possible, we want to hold conditions constant across different IVs, that's independent variables, if we have more than one independent variable, and across different levels of each IV. And here in yellow, we're going to try to remind ourselves of this distinction that many students struggle with, and that is the distinction between having more than one independent variable and having multiple levels of a given independent variable. So let's just remind ourselves first of what a variable is. A variable is an entity that can assume more than one value. In the example that I gave a moment ago, the independent variable would be the variable that was the stimulus duration. It was 10, 20, or 30 milliseconds in duration. That might be my variable of interest. So that would be the one variable, stimulus duration, and it has three levels. The variable stimulus duration then has three values. I could have an interest in a different independent variable. It might be something like the brightness of my stimulus. And you can begin to quantify brightness different ways. We can say that, for example, a particular stimulus might be 100 units bright. Another one might be only 50 units bright, and we can say that we have the brighter or dimmer stimulus. And we can make that a different kind of variable. That would be maybe the brightness variable, and it would be high intensity or low intensity. It might be the two levels of that one variable. But that variable is distinct from the stimulus duration variable. So we can think about independent variables as the variable whose effect we seek to know, and we can very clearly see that any given independent variable would have to have at least two levels for it to be a variable. It could have many more levels than that. So getting back to this statement, we can say that whether we have multiple independent variables or even a single independent variable that has multiple levels, we want to hold conditions constant to the extent that that's possible. Okay? When we can't hold factors constant, we resort to balancing. So just a word before I move on to that, and I'll say just a little bit more about balancing. Once we've held the factors constant, or these alternative plausible explanations constant across our independent variables and across the levels of a given independent variable, then we can eliminate that potential explanation as a plausible explanation for any effects that we might observe. So holding factors constant is very important. When we can't do that, and sometimes for practical reasons we can't do that, then we resort to balancing, which is evenly distributing the uncontrollable variations across all experimental conditions. Okay, the most important factors that cannot be held constant in most psychological research would be what we call participant variables. And you might have an idea about what we mean by participant variables. I'll give you some examples, just to make sure that it's really clear. In our studies, we are going to have lots of participants, and these participants might differ in any number of ways. It might be the case that they differ, for example, in gender, or they differ in their background experiences. They might differ in their ages. They might differ in their relative motivation levels. They might be sleep deprived to different amounts. They might be variously motivated to do well or to not do so well. These are factors that you and I cannot directly control, so what we have to do is resort to some kind of balancing, where we will acknowledge that these differences do in fact exist, but we try to evenly balance them out across the various conditions that we have. Okay? All right, so there are a couple of different ways that we can do this. One way is through random assignment, a procedure by which each participant has an equal opportunity of being assigned to each different experimental condition. And here we're going to make sure that we clearly convey to you the distinction between random assignment on the one hand and random selection. So we'll ask you first to see if you can stop the video and recall what that distinction is, random assignment versus random selection. Okay, welcome back. Hopefully you were able to muse about something like this, and that is, in random selection, we're simply trying to determine who gets into the study in the first place. You'll recall that we have a population, which is the collection of every member in whom we are interested. All members of a group of interest would be our population, and we would use a sampling frame, an operational definition of our population, in order to pick random samples to bring into our study. Now, if we have every member of that sampling frame getting an equal chance to be selected into the study, then we can say we have truly random selection, and we now have presumably a very representative sample of the overall population that's entering into the study. Once they've entered into the study, there's another decision that has to be made by you as the researcher. And that decision is who gets into which particular condition. These are the folks who are in the study, but maybe not everybody gets into all the conditions. And the reason for that is we might be running an independent subjects design, where any one participant might receive only one level of the independent variable. Okay, so who gets into which kind of condition? Now we're on the issue of random assignment. Who gets placed into this condition versus that condition? In the example that I gave earlier, which of our participants might be assigned to the 10 or the 20 or the 30 millisecond condition? Okay? So if there were two conditions in an experiment, random assignment could be accomplished by something like a coin flip. And you could either literally flip a coin, or you could use something like Excel to help you generate maybe a 1 or a 2 randomly. And if the number 1 happens to come up, then that person would be assigned to condition 1. If a number 2 comes up, then that person is assigned to condition number 2. And we can use Excel to simulate other kinds of coin flips, in air quotes, where we might have many more than just 2 conditions. We might have 13 conditions, for example. And we can have Excel randomly assign any given person who's been randomly selected from the population into one of our, for example, 13 different conditions. Okay? Now, one of the disadvantages in doing that is that if you're using purely random numbers to make the assignment, you might remember that random numbers have this very unique property which is called independence. And what that means is that the coin flip on any given trial is going to be independent of all other coin flips. And a consequence of that independence is that we might get these strings in which many people are assigned to a particular condition without being assigned to other conditions in an evenly distributed manner. You might remember that we can generate strings of heads or strings of tails if we have random coin flips. Now, what's important about that is that sometimes in experimental psychology, we have only a very limited number of observations. And we want to, as fairly as possible, distribute those observations across the different conditions. So it would be perhaps inappropriate to have 30 people in our study, and instead of having 10 people in the first condition, second condition, and third condition, it could be that if you were to use an Excel coin flip, you would have 25 people in one of those conditions, 2 in another, and 3 in another. This would be grossly imbalanced. So we probably, especially when we have lower numbers, we can't rely on a simple coin flip. The independence of random numbers might come back to bite us. So what we do instead is we engage in a type of pseudo-randomization that we call block randomization. And this is a very important idea in experimental psychology, and one that tends to throw some students because it's not all that intuitive. Let's see if we can first identify what this is. Block randomization is a procedure for random assignment requiring that each experimental condition has received one participant, or one observation, before any experimental condition has received a second participant, or a second observation. Now, some people struggle a little bit with that definition. It's a little bit long. We'll see if we can make sure that people understand it clearly in this video. Here's a schematic of what we might be talking about. We'll do block randomization for one block of trials, and a block is a complete set of experimental conditions. Okay, so let's go back to the example that we've been developing, where we have a study, that's a perceptual study, and we show the stimuli for 10 milliseconds, 20 milliseconds, or 30 milliseconds. We'll call those conditions A, B, and C. If I gave you a trial, or a sequence of trials, A, B, and C, I would have a complete set of conditions. You'd have one at 10 milliseconds, one at 20, and one at 30 milliseconds. What we might want to do in block randomization is make sure that we get one observation in each of these 3 conditions before any of those conditions gets a second observation. So if we're using a between subjects design, we might assign the very first participant to, say, randomly C, but then we can't assign a second participant to C until we've gotten at least one in condition B and one in condition A. Having satisfied that complete set, then we can begin anew, and we can shuffle these around and start one more time. That might be shown here, for example, where we might have these 3 conditions, again, 10, 20, and 30 milliseconds hypothetically, and we might put our first participant into condition C, second into B, third into A. Now every one of those conditions has been satisfied once, and then we might start again in a new random order, and here's another random order, and we keep on assigning participants across this scheme, randomizing the sequence in this block random manner. For this set of just 3 conditions, you can see that we have 6 different possible combinations, which is very interesting to muse about the different ways that we can assign people to this sequence. And all the while now we're hoping to control for such things as order effects, and we're making sure that we don't get a buildup in any one of these conditions to the exclusion of other conditions. Okay, so let's see if we can summarize the advantages of this partially counterintuitive block randomization procedure. Here are a couple of advantages. First, block randomization ensures there's an equal number of participants in each experimental condition, and this tends to increase the capacity that we have to find an effect if an effect is there. If we have 30 people and 3 conditions, we can be assured that using block randomization we're going to wind up with 10 in each of these, whereas had we used purely random numbers, we could have wound up with a gross imbalance with a much overrepresentation in one of the conditions and large underrepresentation in another one of the conditions. Okay, so that's one of the nice features. Secondarily, we can say that block randomization automatically controls for time-related variables, and there are a couple of different ways of thinking about this. When we're talking about time-related variables, issues of fatigue and practice effects can rear their heads when we're dealing with a within-subjects design, and something called an early bird effect can rear its head when we're talking about a between-subjects design. Let's say a little bit about each of those in turn. We'll talk about the within-subject case, and this is the scenario where everybody's experiencing all levels of the independent variable. As you might imagine, in the example that we've been giving with 10, 20, and 30 millisecond stimulus presentations, even the 30 millisecond presentation is quite brief, and people might need a little bit of practice before they get to be good at seeing these stimuli shown at such brief intervals. So if we can take advantage of block randomization, what we can say is that maybe you'll get better with practice after you've done 10 or 20 of these trials versus your first few. If we can block randomly distribute the sequence of experimental conditions, then all of the practice effects will be equally distributed across the 3 conditions. Similarly, if you do several hundred trials, as is the case frequently in my own experiments, after a while you begin to fatigue. We wouldn't want that fatigue, that fall off in behavior that's due simply to you getting tired, we wouldn't want that to affect only one of the conditions. We want to have that affect all of the conditions equally. And by taking advantage of this kind of a scheme, we're distributing the practice effects across the 3 conditions, and then through here you're getting to be really good, and now you're starting to fatigue, but we're distributing that evenly for the across the different conditions when we're using block randomization in a within-subjects design. Let's see if we can now move over to talk about the early bird effect in between subjects designs, and we'll just remind you of something that you might have heard about in Intro to Psych, but maybe not necessarily. The early bird effect: participants who volunteer early in the semester might be systematically different from those who let things go to the last minute. So we all know people who let things go to the last minute, and many of us have been in Intro Psych classes where we had an experimental participation requirement. And some early birds, or eager beavers, will tend to come in the first weeks of class, and they will tend to be very motivated and participate in our experiment. And then as the semester rolls on and things are piling up toward the end of the semester, some students will come into our study only because they have to and they've let things go. And they might be a little bit distracted toward the end of the semester, partly because they're fatigued, partly because so much is due with final exams and so forth, and their performance might be measurably worse than those at the beginning of the study, even though they're getting the same stimulus conditions. Well, if we didn't use block randomization, then we might have a bit of a confound here if it were the case that we had people in the early part of the semester who were highly motivated all landing in, for example, condition A, and people who were less well motivated and a little bit more stressed landing in condition C. We'd like to be able to distribute those more evenly across the semester, and we can take advantage of block randomization to do that, to essentially rule out this this effect that we might have that we call the early bird effect. So block randomization can control for time of year effects or time of semester effects and so forth. In time of year effects, you might imagine that we have such things as seasonal affective disorder, and some people during the winter months are feeling a little bit down, maybe because they're not receiving as much sunlight. We wouldn't want all of those folks to land in just one of our 3 conditions. We'd ideally like to distribute that particular form of variation evenly across our 3 different levels of the independent variable. Okay, let's see if we can go on and say something about 2 topics that you might remember from Intro to Psych, but since we're talking about experimental control today, it's important that we remind you of this terminology. The first is one that you might know called the placebo, a substance or condition that has no effect and is used to control demand characteristics. And you might remember that we've talked about demand characteristics before, and these are cues that affect the participant's behavior. Frequently, participants are very sensitive to what's going on in a study, and they might sense that you want them to behave in a particular way, and they'll try to, in many cases, match your expectations. And so their behavior is changed based on some cue that they've picked up from the environment. This is the nice thing about a placebo effect, and in my own studies, I've been able to find that I can offer placebos to control for some demand characteristics. As I've mentioned before, I've been interested in how quickly people can see these briefly flashed stimuli, and I'm occasionally interested in the effect of caffeine. So I'll have some of the people in my study take some caffeine. They might drink a Coke or a Pepsi, for example, and I'll have other folks on a placebo, which might be a decaffeinated Coke or a decaffeinated Pepsi. As you're drinking one of those two possible soft drinks, you might not know which one you're drinking, but you know that you're taking in something that could be caffeinated, and hopefully this will control for some of the changes in behavior that we might expect just based on what you believe about how these soft drinks might be affecting your behavior. So the placebo effect then will offer some control against these demand characteristics. A related idea, but not exactly the same idea, is this double-blind procedure, a procedure in which the participants and the data collectors don't know who's received which level of the independent variable. So not only is it the case that the participants don't know whether they've received, for example, a real Coke or a real Pepsi versus a decaffeinated Coke or a decaffeinated Pepsi, they don't know, but also the people who are collecting the data and making the behavioral observations don't know. And what's nice about this is it controls for demand characteristics, as was true for the placebo. It also controls for expectancy effects. That is, when researchers are making observations, and for example, they have to classify a particular behavior as landing in this category or landing in this category, sometimes there are ambiguities. And it isn't entirely clear whether to classify an observation into one category or the next. Surely, the researchers themselves are people and they have their own biases, and they might be inclined to classify behavior relatively favorably in the caffeine condition if they know the participant has had some caffeine. And so to control for that expectancy effect, we can take advantage of a double-blind procedure. Okay, so before we wrap up, there's one other point I would like to make. I'd like to bring us back to this notion of confounds. And the reason why I want to do that is through the years, when I've come to this point in the semester, I find that we talk about drawing inferences from experiments and trying to draw causal inferences in particular. And students learn the word confound, and we can define a confound as systematic covariation between one factor and the independent variable. And we can define a confound that way. Students tend to use confounds a little bit inaccurately. So I want to conclude today's session by showing you a Venn diagram that contains a relationship between confounding variables and other kinds of challenges or problems that we might experience in the research that we do in psychology. So let's break out of this, and we'll show you a Venn diagram that I hope you'll find interesting. Welcome back. We're going to continue our conversation, and we'll be talking specifically about confounding variables. And I think this is important because so many students use this term incorrectly. The students, as they are learning about research methods, frequently will understand that a confound is a problem in research, and they're correct. A confound is a problem in research. But not all problems are confounds. And that brings us to this Venn diagram where we have, in this large circle, the entire collection of research problems. Within that, we have a very special type of research problem that we call a confounding variable, and we might define a confounding variable as a variable that systematically covaries with the independent variable. Recall, too, that the independent variable is the variable whose effect we seek to know. And it might be the case that we have some covariation between some so-called nuisance variable and the variable whose effect we seek to know. So I'll develop a sample or an example that's similar to the one that we had earlier, and this will now give us a clearer idea about what a confound might be, and we'll contrast that with other kinds of problems that are not also confounds. So let's start out with a scenario that it presupposes that you and I are sports psychologists, and we've been hired by a sporting company that makes baseballs to determine whether the color of a baseball might affect its hittability. As some of you might know, American baseball typically features white baseballs, but it wouldn't have to be that baseballs are white, and it might be the case that the game becomes more interesting if people are able to hit the ball more effectively, and this might be a perceptual matter of whether certain colors are more easily hit than other colors are. So we might start out with red baseballs or green baseballs or blue baseballs, and we might test the hittability of those three different levels of our ball color variable. In this study then, the independent variable would be ball color. It would have 3 different levels. As I alluded to earlier, we could engage in all kinds of block randomization to try to evenly balance who gets exposed to the red, green, and blue colors, or we could concoct an experiment that has a confound for illustrative purposes. So let's do that. Let's say that we want to test the hitability of red, green, and blue baseballs. And let's say that we administer the red baseballs to a random sample of students who are batting at 6 in the morning. And then we have a different sample of students who are going to be batting in the afternoon, and they might be hitting the green baseballs. And then the blue baseballs might be hit in the evening by yet a third independent sample of students. What we can say here is, yes, we have 3 different levels of our independent variable, and different groups have been in each of those, or across those different levels, but we also have a confound, which is time of day. That is, remember that the confounding variable is a variable that systematically covaries with the independent variable. And here we had morning, noon, and night covarying with red, green, and blue. So we'd have a confound because of the systematic covariation between the nuisance variable, time of day, and the variable of interest, ball color. So that would be a problem, and it wouldn't just be a problem, it would be a confound. Another example of a confound. Let's say that I had all the really good hitters assigned to the red condition, and I had all the really bad hitters, like me, assigned to the blue condition. Unsurprisingly, we'd have more hits in the red condition than in the blue condition, but not because of ball color, but because of some idiosyncrasy of the hitters. Okay, that would be a confound in this particular case. It's not just a problem, but there would be a systematic covariation between ball color, which is the variable of interest, and the player's ability, which is really the nuisance variable that we would rather distribute randomly to get rid of this confound. Now let's think about a different kind of error that we might have that would not also be a confound. One possibility is that maybe we've taken a sample of students from a college, and maybe college students are pretty misrepresentative of people in general, at least with respect to baseball hitting. I don't know whether that's really true, but let's just pretend for a moment that that really is the case, that baseball playing or baseball hitting among college students is really quite different than it is in the broader population. We would have a lot of trouble generalizing appropriately from this college sample to the wider population, and that would be a problem. But it wouldn't be a confound. Okay? It would clearly be a problem with the research, but it wouldn't be a confound. Another example, let's say that we're using a hitting machine that tends to deliver the baseballs at a relatively slow speed, maybe something like 15 or 20 miles per hour. Most professional baseball players, or even not so professional baseball players, would be hitting baseballs that move much more quickly than that, much more quickly than 15 or 20 miles per hour. So we would now say that this study, again, is highly misrepresentative because the pitching speed is so slow. That would be a problem, but it wouldn't be a confound. And the reason it wouldn't be a confound would be because we would have really slow pitches in the red condition and really slow pitches in the blue and green conditions. So the fact that there are slow pitches is problematic in general, but it isn't specifically a confound, the scenario in which we have some variable, like ball speed, systematically covarying with the independent variable. So I hope that helps you to appreciate the distinction between research problems in general and the very specific case of a confound. And I hope this will help you to use those terms more appropriately. So as always, please make sure that you jot down what was very clear about this presentation and what was less clear, and bring those questions and comments to class. See you in class.